Hello students, I hope you all are revising the concepts every day and you are solving the worksheets parallelly. So today we'll be discussing the homework worksheets that have been assigned to you, right? So let us quickly start the discussion. Now during the discussion we'll be analyzing the questions, we'll be analyzing the concept, and even we'll be learning the strategy how to answer the correct question. Fine. So let us quickly start this worksheet discussion session there. Moving to the first question. Let us read the first question. So here first we will be solving the botany questions followed by the zoology questions there. So let us discuss the first question from the worksheet. Okay, so the fluid nature of the membrane is important from the point of view of the functions. So here as in the chapter cell, right? So you all have read about the cell organelles, the structure of the cell membrane, right? Now here they are asking, now with respect to the cell membrane, what are all the functions that it carries out? Okay, so what are they? So the cell division and the cell growth, okay, the endocytosis and the secretion part, the formation of the intercellular junctions, and all of the above. Now while discussing these topics, so what exactly we have gone through? So it helps in all of the given above processes. So what exactly will be the answer? The answer will be all of this. Fine. Coming to the next question. Now here they are asking the color of the flower petals, right? So here we see the colorful petals, right? Now what exactly is responsible for that color of the petal there? Okay, so what exactly is responsible for the color of the petal there? Now is the leucoplast, right? Now it's a cell organelle, right? Okay, next it's not the leucoplast, it's the chromoplast. Okay, it's not the chloroplast, because chloroplast is what? The green in color, right? It's not the mitochondria because it is the cell organelle. This is clear? Yeah. So here for example, when you are solving the question, do read out the question, and if you don't know the correct answer, what you do, you eliminate the options. So in case you eliminate the options, what will happen? Okay, so here you can quickly analyze with the remaining options and mark the correct answer. Fine. Let us move on to the next question. Now here they are asking the plant cell differs from the animal cell by. So here while discussing the structure, while discussing this chapter cell, so here we have discussed about what? The differences between a plant cell and the animal cell, right? So here what exactly is the, the main difference? Like for example, both the cell, they have the cell organelles, right? Both the cells, they have the cell organelles, but what is present in excess and what is absent there? For example, if you see the plant cell, if you see the plant cell, if you see the animal cell, right? If you see the animal cell, now what are the major things? Now what is commonly present in both? Okay, what is commonly present? Commonly present, you can say the cytoplasm, right? You have what? The mitochondria. Fine. The vacuole, but in the case of this one, it is little big vacuole. Fine. Now next we have this Golgi body, right? Then we have this endoplasmic reticulum. Now we have the cell membrane, which is common in both of them. Now what is excess? Now for example, if you see the plant cell, you also have a cell wall. You also have a cell wall, but here the cell wall is absent. Here the cell wall is absent. So what will happen? Now for example, if you place the animal cell, in the critical thinking question, they will be asking you, if you place an RBC. Okay, so if you place an RBC, that is a red blood cell, if you place the red blood cells in the water, okay, so what will happen? So here, due to the osmotic differences, the cell will be swelling up, and that would be increasing and becoming the maximum size, and that causes the lysis of the cell. What exactly does it cause? The lysis of the cell. Why? Because here it will be taking up the water there. But whereas in the case they have the cell wall, right? Here the cell wall is absent. Apart from that, what exactly? So here you can say in the plant cell they have a large vacuole, yeah, but here they have the small vacuole. Apart from that, they also have this plastids. Okay, now here in this case you don't observe the plastids, right? The plastids are absent. The plastids are absent, and what you can see, the centrioles are present. The centrioles are present, but here the centrioles are absent. Now what they are asking in the question, now presence, the plant cell differ from animal cell. How exactly? The presence of the vacuoles is both is present, both of them are present. Presence of cell wall and plastid, yes. Now absence of the cell wall, no, cell wall is actually present. Absence of the chloroplast, that is also right. See, even if you don't know the answer, what you can do, you can eliminate the options. Na? So it is a plant cell, so for sure it will have a chloroplast. So one option eliminated, right? Then the presence of the vacuole, the vacuole would be present, right? That is also a common thing. You can eliminate it out. Now they are saying absence of cell wall. Here they are saying presence of cell wall and plastid. So if you know the concept clearly, you would be directly marking up and going for the second question. Got my point? Next, let us go to the next one. Now, which of the following cell organelles are enclosed by a single membrane? They are saying, see, the cell organelles, they are bounded by what? They are bounded by the membranes. Few membranes are, the single membrane, and the, most of them are the double membrane, like your, the mitochondria. Fine, right? So here, what do you say? Apart from that, you have the other cell organelles, like you have the chloroplast. Fine, they have the double membrane, whereas you have single membrane in some structures like the vacuole, and the the layer is called, the membrane is called the tonoplast. Yes, in the similar way they are asking, they have given the few of the cell organelles, and they are asking you which of the cell organelle is having only single membrane. Got my point? Yeah, so I think it's very easy. So here you know that what are double membrane structures, right? So see, mitochondria is double membrane, chloroplast is double membrane, nucleus is double membrane. So you can do odd one out, and then you can just directly mark the answer. Got my point? Yeah. Next. So you should develop this strategy of marking the answers. See, whenever a question comes in front of you, even you feel that you don't know, the right answer, and you have a small confusion, but like little of the concept. So what you can do, you can just work wisely, and then you can eliminate the options which you feel is like incorrect. Okay, you're confident, you're not confident what is the correct answer, but you are confident that these options are wrong, so eliminate those options. Okay, so remaining options, whatever you have, okay, so what you do, you just apply some little logic, whatever the concept just try to apply it, and then mark the correct answer. So this skill you need to develop, students, because it is very helpful for you. Okay, rather than leaving the answer, you can attempt the answer, right, by using this skill. Fine. Next, going for the next question. Now they are asking what is the common between. So what is common? What exactly is the common between the chloroplast, the chromoplast, and the leucoplast? Okay. So here, what exactly is the similarity there? See, chloroplast, if you have solved the previous questions, so what exactly did we study? There was a question. In the question they were asking, the color of the petal is because of what? Okay, the color of the petal is because of the what? The chromoplast, right? And chloroplast. I think from the grade first you are studying that why are the leaves green in color. So you are studying because of the pigment called chlorophyll, which is present in the chloroplast, right? So there is two similarity. Okay, even you don't know the leucoplast, leave it. But you know the two of them, right? So what is the similarity? That is, they are something related to what? The color. So they, because of the presence of these organelles, okay, they will be what? The color will be there. Fine. Now here what exactly is there? Because of the pigments, right? Because of the thylakoid and grana is only present in the chloroplast, right? Storage of starch, proteins and lipids, no. Ability to multiply by a fission. Okay, so all of them three, so they are saying similarity. Okay, so we don't want how exactly they are different. See, but here I can say presence of the pigments, Fine. Okay. Next, the presence of thylakoids in grana, it's only like few, and presence of the starch, lipids, and the protein. Now, what exactly is the common in three of them? What exactly is the common? That they multiply, the ability to multiply by a fission. Next, the structure that are formed by stacking of organized flattened membranous slag in the chloroplast are called. Okay, so if you know the structure of the chloroplast, okay, when this topic was, when we have seen, so this is what a thylakoid. So here they will be what? Stacking over each other. Okay, so in the cell organelle, you have the cell organelle, right? So here you have the structures. Now these thylakoid, right? This is your stoma, the connecting stoma lamella, right? This is your thylakoid. And the stack to form the grana, right? This is your stroma lamella. Okay, fine. And this is what your structure of chloroplast. The structure of what? The chloroplast. Now here they are asking what? In the structures that are formed by stacking of the organized flattened membranous sac in the chloroplast are called what? Okay, the stacking. So here the answer is what? The grana. Okay, it is not the stoma lamella because it is connecting. It is not the stoma because see this empty spaces here is called what? It is the stoma, right? Now it is not the cisternae because cisternae is present in the mitochondria. Fine. Even if you know the structure, you would be able to answer this question. Next, let us go to the next question. Now here they are asking us the correct sequence. They are asking what is the correct arrangement of lipid molecules in the cell membrane. Okay. So what is the correct arrangement there? Fine. If you know the structure of, you know, the membrane, right? Now how is the structure of the cell membrane? Now for example, right? So here you have, it's a bilipid. Okay. Now here what are these? The head, and these are your tail. The head, polar head, and this is what your non-polar tail. Now the head is projected outside whereas the tail inside. Got it? Fine? Yeah. So here let us read the options, the correct arrangement. Okay, they are saying, what is the correct arrangement of the lipid molecules in the cell membrane? The polar head, which is outside. Okay, that's right. Next, non-polar tails, which are what? Inside. Now next, the non-polar head outside. See, outside is there. The head should be outside. Here they are saying the tail is outside. So this is anyhow wrong. Okay, so here, but they are saying the polar tail, which is inner side, the tail is non-polar. So you can go with the first option, that is polar head, which is outside, and non-polar tails, which is inside. This is clear? Yeah. Next, yeah, the centrioles are present in. Now the cell organelle, the centrioles. Now if you remember differences between the plant cell and the animal cell, okay, so then this question is very easy for you, right? So they are saying centrioles is present in a specific cell. So we all know that the centrioles are present in the animal cell. They are not present in the plant cell. They are not present in the animal and the plant cell. They are not present in both. They are not present in all like cells other than the plant cell. Okay, so it's also a very easy question. Next, what is the function of the cell wall? Okay, so here they are asking the function of the cell wall, right? So here what exactly the cell wall does? It gives a shape to the cell, obviously. Okay, it gives a shape to the cell, yes. Next, apart from that, it protects the cell, yes, it protects the cell. Next, cell to cell interactions, yes. Next, all of this. So as 3 of them are right, the answer is all of this. Next, moving to the next question, what are the chemical composition? So we know the cell walls, right, they are made up of, okay, what exactly they are made up of, they are asking. Okay, so here they are saying what are the composition, what are the chemical composition of algal cell wall. Okay, so here you need to clearly see that they are not asking, you know, on a broader sense, they are specifying that what is the chemical composition of this algal cell wall. Okay, they are specifying what is the chemical composition of what? This algal cell wall. Okay, what exactly? So they have given 4 of the options. I mean, what are they? They have given cellulose, okay, the galactans, the mannans, and the calcium carbonate and chitin. So they have given 5. Now they are asking out of 5, okay, how many of them belong to what? The algal cell wall. Okay, so even if you don't know this answer, what exactly do you know? You know that you know the chitin, where exactly the chitin is present. Now the chitin, like you know the fungi cell wall is made up of what? The chitin. So what you can write down here? In the fungi, okay, the cell wall is made up of chitin, right? It is made up of chitin. Next, you have this calcium carbonate. Now calcium carbonate, fine. Now where exactly the calcium carbonate? You can see in the organisms, mainly, you know, the shell, right? For example, some of, you know, the sponges also, the endoskeleton and exoskeleton are made up of this calcium carbonate. It's a very hard structure, but it is not something for the algae there, right? So here we have what? Here we have this cellulose, then we have galactans, next we have this mannans. Okay, so here it's made up of, so here they are present in what? They are present in your algae. Okay, the algae cell wall is made up of your cellulose, galactans and mannans. So if you check, it is a direct line from the NCERT. Fine, even in your given module, the reso module, it's a directly the line given in the paragraph. So here what you have to do, so whenever you're reading the concept, okay, whenever you're reading the module, so read it properly, right? Highlight the, you know, few of the statements which you feel are more important. Getting my point? So that during the revision time, you can quickly go through it. Okay, so what's the answer? So total 5 are there. So out of 5, how many of them belong? Only 2 of them belong? No, we have 3 there. So only 3 of them belong. It's not the 4, it's not all of this. Next, moving to the next question. Yeah, so here we are done with the in-class discussion questions. Let us read out, let us solve this homework question. Now what is the first question? Now lipids are arranged within the membrane. Okay, so they are asking about the arrangement of the lipids. Lipids are arranged within the membrane with what? Okay, so here what did we study? If you remember the structure, okay, how exactly is the structure? Fine, how exactly is the structure? Right, let us just draw it one more time. So here, these are the heads which are polar in nature. The head which are polar in nature. Okay, so this is the head and these are the tails. Okay, so head, where exactly, what is the nature? It is polar. The tail is non-polar. Where it is present? The tail is present to the inner side, whereas the head is present the outside the environment. Fine. Now let us analyze the options. Okay, let us read out the options. So they are saying polar head towards the inner side. How it is the inner side? It is wrong. Both the heads and tails to the outside. It is also wrong. Head towards the outside and tail towards the inside. Yes, it is right. Both head and tail towards the inside. No, this is wrong. So the answer would be the third one. Fine. It's a very important concept, students. So you can, whoever who marked this wrong, you can quickly open your, materials and go through this concept one more time. Fine. Next, why tails of lipids in the membrane are towards the inner part. So they are asking why exactly. Okay, the tail is present towards the inner part. But what did we study? What is the nature? So the head which is present outside, that is polar. Fine. And then next, the tail which are present inside, that is non-polar. Fine. So the non-polar, they are hydrophobic. And see, for example, if we drew this structure, so quickly let us do the structure one more time. Yeah. So this is something. Okay, right. Now here the non-polar part. Now here this is what? This is the head. This is bipolar. And this is what? The tail, which is the non-polar. What is the tail? That is hydrophobic. What is the head? Okay, it is what? The hydrophilic. Hydrophilic. Okay, so here let us analyze these statements. Let us analyze the statements. The tail is non-polar. Okay, and also protected with an aqueous environment. No, the tail is a polar hydrocarbon and is so protected from an aqueous environment. The non-polar, okay, the tail is not polar, okay, it is what? Non-polar. The non-polar or hydrophobic and tails of the lipids being in the inner side. The lipids, it is the inner side, ensure their protection from the aqueous environment. So here they don't. So here the option number 3 is correct. The tray is hydrophilic. No, it is not hydrophilic. Okay, so and it is not tending because it is hydrophilic, it should be outside. Got my point? So the answer would be the option number 3. Now let us move on to the next question. Now in the question they are asking which of the following statement is incorrect. Okay, so you should be very careful what they are asking. Not incorrect, okay, or not correct. Fine. So here if you miss up at least a little one, then you will be marking this wrong. So here it is incorrect about the plasma membrane. Now the ratio of the proteins, the first statement, ratio of proteins and lipids varies considerably in different type of cells. Yes, that is what? That is a correct statement. This is the correct statement. Next, 52% of the protein and 40% of the lipid are in the membrane of human RBC. Yes, that is also correct. The arrangement of the proteins and lipids is lipid, protein, protein, lipid. No, this is what? Incorrect. Now what is the correct sequence then? The correct sequence should be protein, lipid, lipid and the protein. This is the correct sequence and this sequence is not correct. Okay, the head of the lipid is hydrophilic. Yes, the head is hydrophilic and the tail is hydrophobic. This is also correct. But in the question they are asking the incorrect one. So the option number 3 would be the correct one. Okay, next let us move on to the next question. The molecules in a membrane that limit its permeability are what? What do you mean by permeability? See, permeability is something which will be allowing the few of the molecules to enter into the cell, right? The movement, allowing the movement. So here, see, we have this permeable, we have semi-permeable. Permeability is like fully allowing, right? If they are permeable, it allows all the molecules. It is impermeable, it will not allow the molecule. Okay, next, if it is semi-permeable, few of the molecules are allowed and few of them are not allowed. So here in this question, they are asking the molecules in a membrane that limit its permeability are what? Carbohydrates? No. It is what? The phospholipids. Okay, so they limit their permeability. Next, moving on to the next question, an important model of the cell membrane is widely accepted model. What is an important model which is widely accepted, right? So it is what? For sure, no doubtly, it's a fluid mosaic model. When this topic was being explained, I think even the the fluid mosaic model, okay, was being explained properly. So here, this is a model which was the most popular, right? It is not the unit membrane model. It is the fluid mosaic model, not the Daniel and Davidson model, not the Robertson model. Fine. Next, who proposed the fluid mosaic model? Okay, so who proposed the fluid mosaic model of the plasma membrane in 1972? Fine. So here the scientists who proposed the fluid mosaic model of the plasma membrane are the Singer and the Nicholson. Fine. It is not the Robert Brown because it's related to the nuclei. This is the Golgi apparatus, not the Robertson as well. Fine. So the answer is what? The option number 1, students. Next, which of the following statement is not true? See, I told you, right? So you should look for such words. They are asking not true. Okay, means incorrect statements about the cell membrane. Okay, now next, it is present in both plant and animal cells. Okay, so is cell membrane? Yeah, it is present in both. But what is not present in the animal cell? Cell wall is not present. Okay, but here cell membrane is present in both of them. So no doubtfully this is what? The true statement. They are asking us not true. Lipid is present as a bilayer. Yes, this is also right. Proteins may be peripheral or an integral part of it. Yeah, it's a directly line from the paragraph if you read the paragraph, thoroughly. Next, carbohydrates are never found in it. No, it's not like that. They are found. Okay, so the option number 4 is not true. Okay, so it is the incorrect option. Fine, students, let us go to the next question. According to the fluid mosaic model of the cell membrane, the proteins are located. So they are saying according to this model, okay, where exactly the proteins are located. Fine. So where exactly the proteins are located? It is a continuous layer over the outer surface of the membrane, right? It is a continuous layer of the inner surface, right? It is a discontinuous arrangement of both the surfaces as peripheral proteins on the interior and the membrane of the integral proteins in the middle of the membrane between the lipid layer only. Okay, so it is what? A continuous layer over the inner surface. Next, moving on to the next question. According to the model concept, the cell membrane is what? The cell membrane is fluid. It's a fluid mosaic model. The fluid mosaic model explains what? It explains only the structural? No. Only the functional? No. Both the structural and functional aspects of the cell membrane. Got my point? Okay, so with this students, we have completed the botany part. Now let us quickly solve the zoology homework questions. Fine? Yes, so let us move on to the first question. Now here they are asking what is an incorrect statement. So select the incorrect statement. Right. So a person of O blood group has an antigen A and antibody B. So let us quickly, write down the ABO blood grouping. Fine. So we have this ABO blood grouping. ABO blood grouping. Next. Yeah. So here what we have the blood group. The blood group, the antigen, the antibody. Okay, Now where these antigens are present, the antigens are present on the cell surface, the antibodies are present in the plasma. They are present in the plasma, right? Okay, so here the blood group A, why it is the blood group A? Because of the presence of the antigen. What antigen? The antigen A. So they have this antigen. What is an antigen? The antigen A here. Okay. They have, because the antigen A, and what is the antibody? The antibody B, the contrast. Why it is the blood group B? Because the presence of the antigen B and the antibody A, because of the contrast. Why it is the blood group AB? Because of the presence of both the antigen A and B, and here no antibodies in the plasma. No antibodies, right? Next, why it is called the O blood group? Because of no antigens, and how many antibodies? They have both A and B antibody. Okay, so this is a generalized concept. Okay, now let us analyze the options. They are saying a person of O blood group has anti-A and anti-B antibodies in the blood plasma. Yes, obviously that is what the correct, but they are asking the incorrect statement. Okay, so this is the correct statement, right? This is the correct statement. They are asking for the incorrect statement. Now person of the B blood group can't donate the blood to the person of A. So they are saying B cannot donate to A. Why B cannot donate to A? Because donate person to the A blood group. Okay, so see what is present. If the B is donating the blood, what is present here? Antigen B on the B RBC. Now when it goes into the A, what happened? Antibody B is present in the blood group A. So here what happened? Here the antigen antibody reaction will happen and there would be a clumping, right? So here it is true statement only, but they cannot donate the blood. The B cannot donate the blood to A and vice versa. Fine. So B can donate the blood to what? It can donate the blood to the B blood group and the AB blood group. Fine. So this is clear. Yeah, so this is also the correct statement. Next, the blood group is designated. They are saying the blood group is designated on the basis of presence of antibody. No, it is not the antibody, but it should be what? It should be the antigen. Okay, so here the correct answer is what? The incorrect option is the option number 3. Let us analyze the fourth one. The person of the AB blood group is a universal recipient. Yes, is it? Okay, so yeah, it is a universal recipient because A can donate the blood. Okay, A can give the blood to the AB. B can give the blood to, AB, even AB itself and the O. Why the A can give? Because antigen A is there. Okay, why they can give to all? Because no antibodies are present in the plasma. Now as no antibodies are present in the plasma, no matter if all the 4 blood groups donate the blood, as there is an absence of the antigen at the time, what would be happen? Okay, so they would be quickly adapting to it or taking it up. There would be no clumping and anything. That's the reason they are what? The universal recipient. Whereas the universal donor, who are the universal donor? The universal donor are the O blood group. Getting my point? Next. Yeah. Okay. Next question. The fluid present between. What is the fluid which is present between the blood capillaries and the cell membrane through which the nutrients, metabolic waste exchange between the blood cell is called? Okay, what exactly it is called? It is called what? Your lymph there. Okay, it is called what? The lymph. Next, in some human beings, clotting of blood takes a long time due to what? So here, when we were reading about the blood, okay, so the blood is made up of what? The formed elements and the plasma. In the formed elements, what you have? You have the RBCs, you have the WBCs, and then you have what? The platelets, right? So here the platelets or the thrombocytes, they play a very important role in the clotting mechanism, right? The blood clotting. So even we have discussed about the mechanism of the blood clotting there. Okay, I hope this is clear. So what's the answer? It is the blood platelets. So many of you might have confusion that why it's a vitamin K. So it increases the clotting, but there is nowhere related to. So here they are asking in the question, blood takes a long time. Okay, blood takes short time if you give the vitamin K injection. It will be enhancing this process overall. Okay, so the answer is what? The blood platelets or the thrombocytes for sure. Next, a person with a blood group AB has. Okay, so why exactly, the blood group, on what basis we are designating the blood groups? If the RBC is having A antigen, then that is a blood group A. If the RBC is having the B antigen, then that's the blood group B. If the RBC is having both the A and B antigen on the cell surface, that is called what? That is your AB blood group, right? If they have no antigens on the cell surface, that is what? That is the O blood group. Am I right? Now here they are asking the person with the blood group AB, right? What exactly they have? They have the antigens, but where exactly the antigens are present? This is your RBC, right? This is your RBC. Over your RBC you have the antigen. Okay, over them you have the antigens. So here you have both the antigens. What antigens? Antigen A and antigen B. That's the reason it's called the blood group. It's not the plasma. No antibodies on the RBC surface. Obviously there is no antibody, but that is not, it is the one. Next, both antigen A and B on the RBC surface, that is right. Both antibody A and B in the plasma. Okay. See, if they are having antigens, just imagine in the plasma they are having antibodies, both antibodies, what would happen? Okay, that would result in the clumping, right? So here what will happen if both of them are there? The antigen will bind with the antibody and results in what? Results in the clumping, right? So here what will happen? So here this is not possible. Okay, so when we discuss only at the time we tell. So in the AB blood group we have both antigen A and antigen B on the cell surface, but we have no antibodies in the plasma, which is completely opposite to the O blood group, because in the O blood group it is called O because no antigens are present on the cell surface, and in the plasma you have the antibody A and B. Got my point? Yeah. Next. So they are asking if antigen A is present on the RBC, then the blood group of a person is. Very easy question, students. Okay. So even you need not to think about it. Okay. So it is what? The A blood group. Next, blood group antigens are found in the HB molecule. Is it the HB molecule? No, right? It is not the HB molecule. Fine. Because what is HB? It's the hemoglobin there. Fine. It is rather what? Found in the plasma protein? No. It is found on the RBC? Yes. It is found on over the WBC? No. It is not found over the WBC. Next, the person with and blood group with are the universal recipient and the universal donor. Okay. So they are saying who are the universal recipient and who are the universal donor. So even if you know the concept well, so here they are asking RH also. In general what you know? In general, you know the universal recipient is AB. In general, you know universal donor is O. Okay, fine. Now here let us analyze the options. First, you know the recipient, that is AB. So here it is O and O given. Eliminate the option. Right? You know the recipients are AB, right? You know because of what? Why they are recipient? Because they don't have any antibodies in the plasma, right? So that is the only thing. I think you need not to write one more time everything. Fine. So for example, here we have this antigen and then we have the antibody. Okay. Okay. Blood group A, you have antigen A, antibody B. Blood group B, antigen B, antibody A. Blood group AB, antigen A and antigen B, you have no antibody. O, you have no antigens, but you have antibody A and antibody B. Okay, so they are saying, so you eliminate out of the two options. What is the here remaining? AB negative and AB positive. Fine. So here see, if it is AB negative, If you are going with the first option, AB negative, it is a recipient. So what did we study in the RH incompatibility? If you know the RH incompatibility, okay, so we studied about the two cases. What was the first case? Can RH positive donate the blood to RH negative? Second case, can RH negative donate the blood to RH positive? Now what are these RH actually? These are the antigens which are present on the cell surface. For example, students, this is your RBC. Like your antigen A and antigen B, you have one more antigen called RH antigen. Now this is from the rhesus monkey. Okay, we spot the same. That's the reason it is named what? The RH. Now just imagine you have this RH antigen already in your body. Okay, so here what will happen? Okay, if the RH negative comes, so here would there be any effect? No, because negative is what? See, if this is present, then this is RH positive. Okay, if there is no RH antigen, then there is what? RH negative. So for example, let us do it for the A blood group. So this is, okay, this is antigen A, this is antigen A, okay, and here you have, let me use, okay, this is the antigen A, and let us use this blue color for your RH. Okay, now what is this, students? Now here this is, you know, the blood group is A and it is positive because the RH is present. Now this is what? This is A negative. Why A negative? Because the RH gene is absent. Fine. Now this is present over your RBCs, right? It is present here over your RBC. Yes or no? Now just imagine. Now here there is a blood transfusion, means negative is going into positive. Any difference will happen? Any difference? No, right? Yeah. So the negative can donate the blood to positive. Now just imagine if the positive is donating the blood to the negative, at that time what will happen? The RH antigen will go into the body. The body will start producing the antibodies and kill that RBC. So the first case is not possible. Getting my point? The first case is not, because the body will start producing antibody against it. So if it is a universal recipient, should it be positive or should it be negative? Okay, it should be positive. So here the first option is given negative, so that is wrong. So here the first option is given what? The AB positive, and it is O negative. Why O negative? You are a donor. Okay, if you have RH antigen, can you donate? Okay, so even if you donate by having a blood group O positive, because of the RH antigen, it can cause the incompatibility. That's the reason the O negative is the universal donor but not the O positive. I think you have now understood this concept very, very clearly, right? So here please mark it. So you see on a broader spectrum, okay, if you can say just the blood groups, the O is the universal donor, the AB is the recipient. But if they are asking along with the compatibility, the RH compatibility, then AB positive is what? Here AB positive is what? The universal recipient, whereas the O negative is the donor. Getting my point? Okay, so this is clear. Let us move to the next question. Fine. Now here they are asking ABO blood grouping is based on. Very, very easy question. It is not the surface antibodies. It is because of the surface antigens but not on the WBC. It is because of the surface antigens present on the RBC. But it is not because of the plasma antigens. See, not antigens are not present. Antibodies are present in the plasma. Right? Next. Or antibody is absent in which blood group? Tell me which blood group. So if the antibodies are absent, means they need to have both the antigens. So if you don't know, okay, if you don't know in which of the blood group you don't have any kind of an antibodies, okay, there you know that they have both type of antigens. So just think in which blood group you have both antigen A and both antigen A and B. Okay, so that is obviously which blood group? That is blood group AB. So in blood group AB you have two antigens, that is antigen A and antigen B. So you don't have any antibodies in the plasma. Got my point? Because if the antibodies are present in the plasma, then what will happen? That results in the clumping. Okay, next, blood group can receive, blood group A can receive the blood from which blood group? Okay, so here let us quickly do it. Fine. So here you have the blood group A. So what is an antigen? Right. What is the surface? Okay, here we have the antigen. Here we have the antibody. Okay, you have this A, you have this B, you have AB, you have the O. Okay. Fine. Okay. Now this is your what? The antigen A. This is your antigen B. This is your both the antigen B, and this is your antigen A, and here you have no surface antigen. Okay, so here this is what? This is your antibody B, this is your antibody A, here this is your antibody A and B, right? Here you have what? No antibodies in plasma, right students? We don't have any antibodies in the plasma. Now let us quickly understand how it is. Now they are asking A can receive the blood from which blood group? Okay, so who can donate the blood to the A? Now just imagine, can A donate the blood to the A? Can A donate the blood to the A blood group? Yes, can A donate to A? Yes, they can, because see, what exactly we need to take? We need to consider the, is there any clumping being observed after the blood transfusion or not? So here antigen A, if it goes into the antibody, in the plasma, what will happen? There will be no clumping. So yes, A can go. Now what about the B? Can B donate the blood to A? Now if B is donating, now what is moving? Now this will be moved. This will be moved into the plasma. So here what is present in the plasma? Antibody B. Now what is present on the surface? Antigen B. So would they be a clumping? Obviously they will be a clumping, right? So that's the reason it cannot donate. Fine, so B cannot donate. Can AB do that? Let us see. When AB is going, so this cell with both antigen A and antibody, antigen A and antigen B will be going there, right? So at that time what would be happening there? So at that time, can you see here? Yeah. So at that time there will be also what? The clumping. Next, if it goes here, the partial clumping would be observed. If O goes, O is having no surface antigens. So at that time what would be happening? At that time there would be no clumping. So here the reason, the answer is what? The A and O. Okay. But A can donate the blood to the AB, that's not a problem, but it cannot receive it from it. Getting? Next, a person. Now we are done with the in-class discussion questions. Let us quickly do the homework questions there. Fine. Now a person with RH negative blood group. So they is saying a person with RH negative should not receive the blood from what? Okay, so an RH negative can receive the blood from RH negative, that is right, they can receive the blood. They can also receive RH positive. No, what will happen if RH positive is there? Okay, so here if it is receiving the blood from the RH positive, then at that time what would be happening? The body would be producing the antibodies, the anti-RH antibodies, and here it would be killing up those cells. Okay, that's the reason what will happen, so they need not to receive it. Next, you also observe this case in the chapter body fluids and circulation that you all will be reading, in the grade 11. Fine. So you will be studying about what a situation, okay, a condition which arises due to this RH incompatibility called what? The erythroblastosis phytolysis. Right. Because of this condition, what will happen? Because of this RH incompatibility, the mother, okay, so here the mother blood group and the baby's blood group here, because of this RH incompatibility, this condition would be arised. So you will be reading in detail about this particular thing. It's a very interesting topic in the chapter body fluids and circulation. Next, O negative person, yes, they can receive it. A negative person, yes, but not the positive one. So the option is what? The option number 2. Next, moving on to the next question, which of the following is not a blood group? A is a blood group, B is a blood group. Do we have C blood group, students? No, we don't have any antigen C. Okay, we don't have any blood group name C. Okay, so we don't have anything C, we have O here because there is no surface antigens present on it. Next, let us move to the next question, lymph. Okay, the lymph is a fluid that is similar to, okay, so lymph is a fluid that is similar to what? Now when we were studying, now what did we study? We were studying that blood is made up of the plasma and what? The formed elements, right? Now the formed elements consist of our RBC, WBC and the platelets. The plasma is made up of 90 to 92% of water, 6 to 8% of what? Your protein, which includes what? Your albumin, your globulin, next your inactive fibrinogen. Next you have what? The mineral. In minerals what they have? You have this sodium, chlorine, potassium, calcium, magnesium, bicarbonate. So when we were discussing this topic, so how exactly we get the lymph? Okay, the lymph here, now when the blood is flowing into the capillaries, because of the pressure, from it is moving from the artery to the vein. Okay, so here because of the pressure, hydrostatic pressure, what happens? The fluid would be oozing out. Now that gets collected in between the tissues and this is called the interstitial fluid. Now when this interstitial fluid enters into the lymph capillary and lymph vessel, that is called what? The lymph. Then when the interstitial fluid enters into the lymphatic capillaries, that is called the lymph there. Okay, now we have studied that the composition of the lymph and the plasma, it is the same. Okay, so they are asking the lymph is a fluid that is similar to, okay, that is similar to what? Here the blood plasma. So here the composition is what? Moreover the same. Next, the lymph mainly transports what? Lymph transports the oxygen, yes. Carbon dioxide, yes, because see, for example, here is a vessel. Now here we have the tissues, right? Here we have the tissues. Now for example, here you have the RBC, it is oxygenated, it is giving the oxygen. and in the similar way it is taking the carbon dioxide again with it, right? There is an exchange. So it transports both, that is it transports oxygen to the tissues and take up the carbon dioxide and get it back for the oxygenation purpose. Digested fat, yes. So here if you remember the lacteals, where exactly they are present? They are present in the villi, the finger-like structure which is present in the small intestine. Okay, so they look something like this, right? The villi-like structure, villi. Now inside them you have the lacteals which absorb what? Mainly absorb the fats. Next, the red blood cells. Okay, so the mainly transports oxygen, carbon dioxide, digested fats and not the red blood cells. Okay, so I think this is an option here. I think it should be what? All of this. All of these. So the fourth option is all of this. It's the typing mistake here. So the answer is what? The 4, which is all of this. Next, which of these cells are found in the lymph? Okay, so lymph has a similar composition to the plasma. Now tell me one thing, do we have RBCs in the plasma? We don't have RBCs in the plasma because the color of the lymph, what is the color of the lymph? The color of the lymph is what? It's a colorless, or it is a pale yellow color. So mainly does not have a color like the RBC. So here it is not, RBC is not present in the plasma. We have the WBCs. Okay, we have the protein composition is similar to what? The blood plasma here, right? The mineral composition mainly. Next, it is not the RBC. Platelets, okay, no. It is the lymphocytes, right? But the more number of lymphocytes are found there, fine, and it is not also all of these. Now lymph flows into the blood through what? How exactly does it flow? It flows through the lymph vessels. Next, the function of the lymph nodes is to, what is the function of the lymph nodes? Now what exactly are the lymph nodes? Okay, we have the lymph nodes, right? What are the examples of lymph nodes? We have the tonsils, right? You have the Speyer patches, right? Got my point? You have this tonsils, you have this Speyer patches. Now what are all these? And even you have more of the lymph nodes, where exactly they are present. They are present in the groins and the armpits, right? So more lymph nodes are present over there. Now what exactly they do? Right, now they will be producing enzymes. No, they store the RBCs. No organ will store the RBC. They filter the lymph and fight the infections. Yes, they will be doing it. Okay, next, it is not also what? The transport of the oxygen. Next, the color of the lymph. What is the color of the lymph? See, the lymph is a colorless mainly, but it also a little pale. The answer is what? Colorless or pale yellow. The lymph does not contain. What does the lymph does not contain? Okay, the lymph, does it contains the WBC? Yes, it contains WBC, but they are asking what it does not contain. It does not contain the RBC. It contains the plasma, it contains the proteins as well. So the answer is what? The answer is the RBC. Next, the major component of lymph. What is the major component? I told you the composition of the lymph is similar to the plasma. Now what exactly is your plasma made up of? What exactly is your plasma made up of? 90 to 92% of water. Next, you have what? You have the protein. What are the proteins? You have this albumin. Okay. You have this globulin, and then you have this fibrinogen. Okay. And then we have the minerals. You have the minerals. What are the minerals? You have sodium, chlorine, magnesium, calcium, right? Potassium. Okay, potassium. Next, you have this bicarbonate. So here, what is the major part? The major part is your water. Now, similarly, what is present more here? The water. Okay, it is not the hemoglobin, not the hormones, and not the enzymes. Fine. Okay. Fine students, so with this I hope you all have marked this correctly. So with this we are done with what? We are done with our homework discussion, right? So as we have little more time, let us quickly revise the the chapter circulatory system that we have completed this week. Let's have a quick one shot of this chapter. Okay, this is clear. So we have what exactly are the things we have discussed in the chapter circulatory system, right? What exactly did we discuss in this chapter circulatory system? Now here in the circulatory system, firstly we have discussed about what? The functions. We have discussed about the functions of circulatory system. Circulatory system, right? Then we have types of circulatory system. Okay, then we have read about the components of circulatory system. The components of the circulatory system, which are what? Which are your blood vessel and what? Your heart, which is a pumping organ. Next, we have discussed in detail about the blood, in detail about the blood vessels, in detail about the structure of the heart. Okay, so before that, okay, we also have discussed about what? The ABO blood grouping. ABO blood grouping. Next, we have discussed about the clotting mechanism of blood. Yes, blood. Next, we have discussed about the structure of heart. Fine, we have discussed about what? The structure of, yes, the structure of the heart. Now let us quickly have a one shot of all these topics, right? Now here talking about the first one, which is what? First topic, that is what? The function. What exactly is the function of your circulatory system? Okay, what exactly it does, the circulatory system? So here yeah, the circulatory system mainly, see, we have discussed the circulatory system mainly consists of a circulatory fluid. What exactly is a fluid? That is the blood. Now what exactly it does? Transport of the oxygen to the tissues, transport of the carbon dioxide from the tissues to what? To the lungs. Okay, next it will be also transporting the metabolic waste, fine, transporting what? The nutrients, transporting the hormones, maintaining the body's temperature, that is homeostasis, right? Now all these are what? All these are like maintaining the body's temperature, homeostasis. All these are what? The functions of the circulatory system, right? Now what exactly it is consists of, and how many types? We have mainly two types of circulatory system. We have open type of circulatory system, we have closed type of circulatory system. So what happens in the open type? In the primitive organisms, the open type was observed. In the open type, the blood flows directly into the body cavity. The body cavity is called the hemocoel, right? And the you have the coelomic fluid there. The hemocoel is also called the sinus. So it doesn't mean they don't have a blood vessels, but they don't form a closed network like found in the closed type of circulatory system. In the open type, you have the blood vessel, but they are not properly developed. Okay, so they don't form a closed network or close connection, right? So that's the reason what will happen, the blood will directly enter into the body cavity, that is called your sinus there. Fine, it is observed in the primitive types and mainly it is found in the arthropoda and few of the mollusk. Whereas coming to the closed type of circulatory system, here in this we have a well developed blood vessels. Now from the organ, okay, the blood flows into the blood vessels and transported to the organ there. They are not directly bathed, the organs are not directly bathed into the blood unlike the open circulatory system. Now this is formed in the most developed vertebrates, right? For example, you can take what you can take the chordates, right? So they have this closed type of circulatory system, and that is with few exceptions. Fine. Now next we have the components. Now what exactly the circulatory system consists of? It consists of what? The blood vessels and the heart. The blood consists of what? The blood is made up of two main components, the plasma and the formed elements. The plasma is having the 90 to 92% of the water, 6 to 8% of the proteins, which include the albumin, globulin and the fibrinogen. The albumin helps in maintaining the osmotic balance, whereas the globulin helps in the immune responses and our body immune system. Next we have this fibrinogen, which helps in clotting. Then we have the minerals, which include the sodium, chlorine, potassium, okay, calcium, magnesium and the bicarbonate. Coming to the formed elements, we mainly have three types. We have RBC, WBC and the blood platelets. Okay, the RBCs are also called what? The erythrocytes. The WBCs are also called the leukocytes. Now again in that we are having two types, okay, the granulocytes and the agranulocytes, depending upon the granules, presence and absence of the granules. The granulocytes we have under the basophils, the cyanophils and the neutrophils, and the agranulocytes we have the monocytes and what? The lymphocytes. Okay, so among them the most abundant are the neutrophils, which is okay, the 60 to 65%. Okay, and then we have what? We have the lymphocytes, 20 to 25%. Then we have what? This eosinophils, that is 6 to 8%. Then we have we have this monocytes, 6 to 8%. Then we have this eosinophils, 2 to 3%. Then we have the least abundant basophils, that is 0.5 to and 1%. Got my point? Now here, and what are the functions? Now whenever there is an asthma attack, okay, allergic, due to allergy, at the time the basophil count increases. Whenever there is the parasitic infection or the allergy to some kind of a food, the eosinophil increases in the body. Next, what are the phagocytic nature? The neutrophils are most abundant, they are having the phagocytic nature, fine, whereas the monocytes are also having a phagocytic nature. Now when these monocytes enter into the tissues, they form what? The macrophages. Okay, whereas the lymphocytes, they help in what? In the production of the antibodies, especially the B lymphocyte. In B lymphocyte, the plasma cells. Now blood vessels, we have the arteries, veins and the capillaries, right? Now the arteries connects the blood, they will be carrying the blood away from the heart, and the veins towards the heart, with few exceptions. Okay, now here if you see that they are thick wall, the arteries are thick wall, whereas then they don't have any valves, and the lumen is narrow, whereas the veins, they have what? Thin walls, okay, and they have the valves to prevent the backflow of the blood. Now here the heart will be discussing about the internal structure. Now the ABO blood grouping, it was firstly discovered by the Karl Landsteiner. Okay, so here due to accidentally during a blood transfusion. Now here the anti-ABO blood grouping is mainly based on what? The presence of the antigen. If the antigen A is present, that's a blood group A. Okay, if the antigen B is present, the blood group B. If the antigen A and B both are present, then that is what the AB blood group. In O, you don't have any antigens. Okay, and you have the opposite antibodies in the plasma. For example, in blood group A, you have antigen A, and in the plasma, you have the antibody B. Okay, in blood group B, you have antigen B on the cell surface, and in the antibodies, antibody A in the plasma. In blood group AB, you have both antigen A and B, okay, and then you don't have any antibodies in the plasma. In blood group O, you don't have any antigens on the cell surface, okay, but you have the antibodies in the plasma. Fine. Now talking about the clotting mechanism, fine, whenever there is an external injury, So the tissue and the damaged blood vessels will be producing what? The thromboplastin. Now here this thromboplastin will be triggering and accumulating moreover thrombocytes at the site of injury. This thromboplastin will be undergoing a cascade of reactions and forming the thrombokinase. In the presence of thrombokinase, the prothrombin is converted into what? The thrombin. Now in the presence of this thrombin, the inactive fibrinogen is converted into the fibrin, which is insoluble and forms a mesh-like structure, and thus prevents the clotting. Talking about the structure of the heart, our heart is mesodermal in origin. It is placed in the mediastinum, okay, cavity. Now our heart is 4-chambered, right. Now the upper 2 chambers are called the atrium or the auricles, okay. They are small chambers, and the lower 2 chambers are the large chambers, and they are called the ventricles. Fine. Now here the blood from the superior and inferior vena cava, it enters into the right atrium. Right. Now here these 2 atrium are separated by a septum called the interatrial septum. The 2 ventricles are separated by a septum called the interventricular septum, and an atria and a ventricle is separated in an atrium and a ventricle is separated by an atrioventricular septum. Okay, so the blood from the superior and inferior vena cava is pumped into the right atrium. Then what? The valve opens. Which valve? The tricuspid valve opens, and the blood flows into the right the ventricle. Now from the right ventricle, the blood moves into the lungs via the pulmonary artery. Now in the lungs, it gets oxygenated, and from the lungs, the oxygenated blood is pumped into the left atrium via the pulmonary vein. Now again the valves open, the bicuspid valve open, and from the left atrium, the blood moves into the left ventricle. Now again when the heart contracts, from the left ventricle, the blood is pumped into the aorta. Okay, and again supplied to the different parts of the body. This is how the blood flows in our body. This is how the blood is circulated. Now the valves, when talking about the valves, we have 2 main types of valves. Okay, the valves which are present between the atrioventricular septum is called the AV valves, and the valves which are present, we have one more type, that is semilunar valves. Now the valve which is present between the right atrium and the right ventricle is tricuspid valve, and the valve which is present between the left atrium and left ventricle is a bicuspid valve or mitral valve. And the valve which is present in the pulmonary artery is a pulmonary valve, and the valve which is present in the aorta is what? The aortic valve. Okay, so with this we have completed the circulatory system. Okay, now in detail we have few more concepts that you will be all studying in the grade 11. I hope students you really enjoy studying this chapter. Okay, so that's all, and the next class will be continuing up with a new chapter. Thank you.